Hello, my name is Abdul Kader, and today we're going to be discussing three pairs of orthologous genes in the organism Myothermus ruber. And these orthologous genes code for glycerolipid metabolism proteins. And so the genes of interest are 0537, 1813, and 2759. And so, what are glycerolipids? Glycerolipids are glycerols, and as you can tell by the name lipids, are lipids attached to glycerols. And glycerolipids are vital because they're involved in a lot of different pathways that are related. Um, for example, glycerolipids can feed into glycolysis, they can feed into phospholipid synthesis, and as we know, phospholipids are the building block of cell membranes, and so phospholipids are very important for cell health. Um, we also got a lot of different substrates with a lot of specialized functionalities. We also got fatty acids, which are energy storage substrates, and they're also related in fatty acid degradation. So we have a lot of energy consumption, energy storage, and energy usage out of these glycerolipids. So they're very important pathways to study for health reasons for these energy storage related diseases that are very prevalent in society, just as diabetes, cardiovascular diseases, things of that nature. And that's why we're interested in glycerolipids. And so if we look at the top here at the EC of 2.7.1.165, we see one of our first genes of interest are MRUB 2759, and it's also orthologous to B0514 in E. coli, but we'll get to the E. coli's later. And so what it does is it feeds into glycolysis. It adds a phosphate group to the D-glycerate and goes straight into glycolysis, and bam, you get energy, you get energy usage, and that's why it's of interest. Another protein that we have that we are also interested in is 2.7.107, which is MRUB 1813, and that actually feeds straight into glycerophospholipid metabolism. So, as I stated before, it can relate to phospholipids, which are very important in cell membranes, or it can backtrack and go to fatty acid synthesis, fatty acid degradation, so energy storage. We also have 2.7.130, which is our MRUB 0537, and it's also a kinase protein that adds a phosphate group to a glycerol substrate. And what that does is you can either go to phospholipid or it can go to fatty acid synthesis and degradation. And as we can tell, so these three genes are very vital for this metabolic pathway, and there's a lot of other specialized substrates surrounding these, so there's a lot of other very vital features that they are related to as well. So why are we studying Myothermus ruber, and why are we using E. coli as a control? So Myothermus ruber is a bacterium, gram-negative. It is also an obligate aerobe, so it needs so it performs aerobic respiration, just like humans. So Myothermus ruber lives in extremely hot conditions, and it lives in vents, and it's a very, very red, as we see here in the color of the image, and that's where it gets its name ruber, which is Latin for the word red. And Myothermus, myo meaning low, thermus meaning heat. It gets its name Myothermus because it lives in lower heat compared to other organisms in its phylum. And that's, you put it all together, Myothermus ruber, low heat, lower heat, and also red. And E. coli. And we're using E. coli to compare our paralog genes because E. coli is one of the first organisms to have its full genome sequenced. It's a very simple organism. Its genome's very short, about 4,000 proteins. And because it's so easy to work with in the lab, it's so cheap, it's everywhere. Scientists have been using it because it's such a good model organism. And we are going to compare the genes in M. ruber to the already well-studied, well-documented E. coli genes as a sort of backup to compare and fill in the gaps of information of what we don't know in Myothermus ruber, which is a very understudied organism. And that leads to another question: why are we even studying genetics in Myothermus ruber, this understudied organism? Well, a lot of discoveries can actually arise when we study these organisms. For example, scientists have discovered um, they have discovered green fluorescent proteins when they studied organisms in the sea. And so there's just a lot of proteins that we aren't really that could just arise that could be of very important interest. For example, if an organism we can see has a protein that can digest gasoline, there's a functionality to that, and we can use it for a biotechnology. So when we study these understudied organisms, we get a lot of potential for new advances. And when we were determining what paralogs could be used, we used our KEGG, thankfully through the Kyoto University, and they built a database of pathways, and we compared these pathways in these two organisms. As we see, the left is Myothermus ruber, the right is E. coli, and what we see when it's green, it indicates that that protein is present in that organism. And so our three genes, as we see, 2.7.165, 2.7.1107, and 2.7.130, are all present in both organisms, and that's how we determined that we could use both of these as as potential research for paralogs. So the bioinformatics tools we used BLAST, and it's a alignment sequence tool. And so we used that to compare the E. coli amino acid sequence to the Myothermus ruber to see if there's any similarities, any differences. We found COG hits, which are functional domains of these sequences. And so the COGs just let us know what parts of the protein what parts of the gene did what, and that was also very useful. For cellular localization, which is to determine where the protein is, we used TMH, which told us how many helixes the protein had, whether it was in a membrane, and a helix is in a membrane, so it would tell us if it was in a cytoplasmic membrane, if it was in a cell membrane. And then we had signal P, which was helped us determine if it was a signal peptide, and lipo-P, which helped us determine if it was a lipoprotein. And p-sort-B was very useful as it just gave us a final prediction of where the protein would be located. TigerFam is a database of protein families, just like PFAM, and so they just have an array of different proteins, different genes that you can compare and put your genes to compare to and see any similarities. PDB, or the Protein Database, has a lot of really cool crystal structures and crystal structures are very, very well and thoroughly researched and we use these to compare our proteins to, because the crystal structures are very, very organized and good comparisons to. We used Metasys to find the EC numbers, and the enzyme commission numbers give our nomenclature system that serves to name the enzymes based on their functionality. The KEGG pathway, as we saw before in two images before, just really helps us. It's a database of pathways, and it helped us compare what organism has which enzymes. And so for our first genes results, we have our E. coli gene B4042 and MRUB 1813. Our score was 75.15 bits. And because it was a short sequence, that's a pretty good bit score. We had an E value of about -2 to the -23. So that's a very small number, good indication, not a lot of randomness. We had a COG of 0.818 for both the organisms, so that means that they had the same functional, they had a lot of similarity to the same functional domain, and they were both very, very, very similar to it based on these very low E values. The cellular localization gave us a prediction that it was going to be in the cytoplasmic membrane. That was present for both. We tried to search in the TigerFam, but we actually didn't get any results. As for the PFAM, we got the same PFK-like superfamily, and we compared those two e-values, also very, very small. So that's a good sign. We looked at the protein database, and we got two crystal structures of diacylglycerol kinases. Two different ones, but they have the same functionality, same EC, and they were on the same map, so we confirmed that they are paralog organisms. As for B3926, which is the paralog for 0537, we actually had an e-value of 0 and a score of 608. Both those hits gave us a COG of 0554, so that was a good indication that they had the same functionality. The e-values compared to those COGs were 0 and 0, so that's also a good sign. Cellular localization gave us a prediction that it's going to be in the cytoplasm. And when we looked at TigerFam and compared them, it gave us a 0 value as well. So that was very, very good sign. When we looked at the PFAM, we had a family of carbohydrate kinases for both the organisms. And when we looked at the protein databases, we got two different crystalline structured proteins that were better hits for each respective organism. Both actually had the same functionality, so we still had a good sign. Enzyme commission number, same, meant that they had the same functionality. And as before, the same map reference was used. Lastly, for our MRUB 2759, this one was a little bit more questionable. We actually had a score of 317 and the e-value of 0, which was a good comparison for the blast. Our COG number for both hits was 1929. And our e-values are very, very small, with the E. coli's being 0 and the mRubers being also very small at 4.06 to the negative 137th power. And the predictions led us to the conclusion that it's in the cytoplasm. We had no TigerFam hits, and usually we would be okay with not having TigerFam hits if we had a good PFAM hit, but the PFAM hit actually gave us a domain of unknown function, and for the E. coli gave us a hit of glycerate kinase family. What we should have seen in the PFAM hit was a glycerate kinase family, but that was not present. Potentially this unknown domain could be a glycerate kinase that's just not well sequenced. The protein database did give us a good glycerate kinase, so that was also resourceful. And when we looked at their enzyme commission numbers, both of them 2.7.1.165. So they serve very similar functionalities, but there's a lot of room for us to question. And so this one is a little bit non-conclusive, but still very strong evidence that they could be potential paralogs. And as seen at the bottom, in the same map. And so in conclusion, we can determine that both these organisms are paralogs, and we actually, if you don't know the difference between paralogs and orthologs, we looked for ortholog genes in all these proteins, and there were none present, despite KEGG giving us a potential site for orthologs. We ran the protein sequences against the full genome of the organism, and we couldn't find any we couldn't find any paralogous genes. But regardless, the conclusion is that we had very interesting orthologous genes in two different organisms that are related to the glycerolipid metabolic pathway that is of extreme interest to energy metabolic research. And I'm glad that we used these very, very resourceful bioinformatic tools that saved us a lot of time. Instead of spending time in the lab doing these experiments, we ran sequences on databases and got answers without wasting money on chemicals, without wasting money on research, and especially without wasting time, which is very important in the lab. So these bioinformatic tools are very resourceful in making these conclusions, and I hope you enjoyed my presentation.